We know about glucose and its metabolic fate to make energy and lactate. But there's a metabolic detour that glucose can take, an entirely different path, one that has nothing to do with energy or lactate. It's a lesser known pathway known as the PPP or the pentose phosphate pathway, and it plays a critical role in cancer cell survival. Bear with me for a second. I'm going to nerd out for a second. What did the Imperial walkers do to the rebel base at Hoth in The Empire Strikes Back? They went for the shield generator. What did Iron Man do at the beginning of Avengers Age of Ultron? He went for the shield generator. What did Picard, the Enterprise D, and the crew repeatedly do to disable an enemy? They went for the shield generator. What is the PPP? It is the cancer cell's shield generator. And that's why it's important that we talk about it today. I do hope that that overly dramatic and nerdy intro helps make the pentose phosphate pathway more interesting. That's what we're going to talk about today. So I'm going to start out talking about this obscure and lesser known pathway by starting with this paper that was published in August of 2023, and it's titled The Pentose Phosphate Pathway in Health and Disease. And it says here that the pentose phosphate pathway, PPP, is the glucose oxidizing pathway that runs in parallel to upper glycolysis to produce ribose 5-phosphate and nicotine adenine dinucleotide phosphate, NADPH. Ribose 5-phosphate is used for nucleotide synthesis or DNA, RNA, and NADPH is involved in redox homeostasis, that's the metabolic shield system, as well as in promoting biosynthetic processes such as synthesis of tetrahydrofolate, deoxyribonucleotides, proline, fatty acids, and cholesterol. Through NADPH, the PPP plays a critical role in suppressing oxidative stress, including in certain cancers, in which PPP inhibition may be therapeutically useful. That's why the importance of this video. Conversely, PPP-driven NADPH also supports purposeful cellular generation of reactive oxygen species and reactive nitrogen species for the signaling and pathogen killing. Genetic deficiencies in the PPP occur relatively commonly in the committed pathway enzyme glucose 6-phosphate dehydrogenase G6PD, which is why we check that in people before we start them on oxidative therapies. G6PD deficiency typically manifests as hemolytic anemia, or the breakdown of red blood cells due to red cell oxidative damage, but in severe cases also results in infections due to lack of leukocyte oxidative burst, highlighting the dual redox roles of the pathway in free radical production and detoxification. This review discusses the PPP in mammals, covering its role in biochemistry, physiology, and disease. It says in the paper here, the PPP is a glucose metabolizing pathway that comprises two branches. The input of the oxidative branch, OX-PPP, is glucose 6-phosphate, which also feeds glycolysis. The OX-PPP converts glucose 6-phosphate into ribose 5-phosphate, while making two molecules of NADPH. The non-oxidative branch, non-OX-PPP, reversibly interconverts ribose 5-phosphate with glycolytic intermediates. In most healthy mammalian cells, in unstressed conditions, the PPP exhibits lower flux than glycolysis, for example, 10 to 100-fold. Nevertheless, the PPP carries higher flux than most pathways outside of central metabolism. Moreover, it can be transiently activated in response to oxidative stress during the phagocyte oxidative burst to meet urgent NADPH demands. One function of the PPP is to provide biosynthetic precursors with ribose 5-phosphate, an essential nucleotide building block, and NADPH for the biosynthesis of fatty acids, cholesterol, proline, tetrahydrofolate, and deoxyribonucleotides. From bacteria to mammals, the OX-PPP is the major source of cytosolic NADPH to support these processes. In another video series, I looked at the percent or the estimated percent of NADPH from the PPP, and it's about 40 to 60% of total NADPH production in the cell. However, alternative routes for cytosolic NADPH production exists, including isocitrate dehydrogenase 1, malic enzyme, methylene tetrahydrofolate dehydrogenase, and formidyl tetrahydrofolate dehydrogenase, and can often compensate to generate the required NADPH for synthesis. The PPP also supports cellular defense against oxidative stress, a driver of cardiovascular disease, neurodegeneration, and aging. To prevent damage from ROS and RNS, cells rely on antioxidant systems, including glutathione, thioredoxin, as well as others. Both use NADPH, actually all of which use NADPH to regenerate reduced thiols, and from disulfides. The OX PPP may be uniquely suited for acute antioxidant responses compared to other NADPH production routes due to its having excess capacity that is not used in basal state but stands ready for times of enhanced demand, reserve flux capacity. Indeed, PPP flux upregulation keeps yeast cells alive when they encounter oxidative substances, protects skin cells from the consequences of UV irradiation, and allows cancer cells to tolerate increased oxidant levels during detachment. While often considered bad actors, ROS and RNS also play physiological roles in mammals and are produced by dedicated enzymes that use NADPH as a reductant. NADPH oxidase, NOx, or nitric oxide synthase, NOS, modest amounts of ROS and RNS play signaling and biochemical roles. Larger quantities contribute to immune defense by killing invading pathogens and therefore preventing life-threatening bacterial and fungal infections. In activated phagocytes, the PPP fuels this process, shifting flux away from glycolysis to generate large amounts of NADPH. And this is the first look we've actually had at the PPP. So let's orient ourselves here for a second. So here's glucose, and this is the upper part of glycolysis, like it mentioned here. Lower glycolysis would end in pyruvate and maybe lactate if it's an anaerobic condition or in a cancer context. But anyway, this is the glycolysis pathway. And you see here the first step from glucose is through an enzyme called hexokinase, or HK, and glucose 6-phosphate is the next chemical made. Glucose 6-phosphate can be further metabolized by glycolysis to make energy, or glucose 6-phosphate can be acted upon by another enzyme called G6PD, or glucose 6-phosphate dehydrogenase. And this is where glucose 6-phosphate can enter the PPP. This is the oxidative branch, and this is the non-oxidative branch. This is the branch where NADPH is made, and this is where other things are made. And then these are pretty neat graphics because basically if the cell needs pentose, it will divert glucose off to the non-oxidative branch to make the necessary 5-carbon sugars to then be used for biosynthesis. And then if there's an overflow, it will redirect towards glucose or towards glycolysis, or it'll go through the oxidative branch. And then if there is an oxidative stressor and the demand for NADPH is higher than ribose, it'll go towards the oxidative branch. So what it's saying here is that this is a kind of a finely tuned machine depending on what the cell needs at a certain time. This pathway is present in all cells, not just cancer cells. It's just that cancer cells rely heavily on this to maintain the balance of oxidants to antioxidants. That's also known as redox homeostasis. And here we have basically the pathway again from glucose to glucose 6-phosphate, and then it can either go down glycolysis or it can go to the PPP to make either NADPH or nucleotides or whatnot. And it's just showing this is the increased oxidative flux or decreased oxidative flux. And it's got some of the feedback mechanisms, whether it be NADPH or whether it be ROS or whatnot. So let's look at the next paper that was titled The Pentose Phosphate Pathway and Cancer. And it was published in August of 2014. And it says the Pentose Phosphate Pathway, PPP, which branches from glycolysis at the first committed step of glucose metabolism, is required for the synthesis of ribonucleotides and is a major source of NADPH. NADPH is required for and consumed during fatty acid synthesis, or the making of new fatty acids, and the scavenging of reactive oxygen species, oxidative stress. Therefore, the PPP plays a pivotal role in helping glycolytic cancer cells meet their anabolic demands and combat oxidative stress. Recently, several neoplastic lesions were shown to have evolved to facilitate the flux of glucose into the PPP. This review summarizes the fundamental functions of the PPP and its regulation in cancer and its importance in the cancer cell metabolism and survival. And I pulled this paper not necessarily to read the abstract, because this is above and beyond the scope of this video. I just want to remember where I got this graphic from because I couldn't find the paper that I got the graphic from. But anyway, it's a really neat graphic. So we see glucose here. Okay, glucose is the start of these processes. And depending on the need of the cell, glucose can be fully fermented into lactate by these cancer cells, which takes part in the micro environment acidification, and that leads to immune dysregulation and so on and so forth. We'll kind of talk about that a little bit more in this basic series when we talk about oncometabolites. But lactate is the end product of glycolysis for cancer cells. That's the end fate of glucose. However, if the cell is under attack from oxidative stress, or if oxidative stressors are building up in the cell, as a way to protect themselves from cell death, it will divert glucose to the PPP to make NADPH so that GSH or glutathione is able to be recycled, and we can maintain this redox homeostasis and allow for the cell to survive. It can also go through the non-oxidative branch for biosynthesis, specifically for fatty acids, for amino acids, for nucleotides, to help the cells proliferate or grow. And you can see here that depending on the cellular needs is where glucose will be diverted. And since glucose is used at 10 to 30 fold more than a normal cell, there's generally plenty of glucose around under normal conditions to make this system happen. So that is why it's important to talk about the PPP. That is what makes the internal shield generator function, by making large amounts of NADPH to recycle these antioxidant systems, which maintain redox homeostasis and allow cancer to repel attacks from chemo, from radiation, et cetera, et cetera. I hope this was helpful. If you like videos like this, please like, share, and subscribe. And until next time.